Good morning, NEET aspirants. Today we are going to discuss about cell cycle and cell division. Okay, first we'll start with the introduction. In the introduction part, the cell cycle is divided into cell cycle. The major phases are, be it mitosis or meiosis, the first phase is called M phase. And the second one is interphase. Second one is what? Interphase. Okay. Now, this M phase is divided into two phases, two subphases. What are the two subphases? What are the two subphases? Yes, first is karyokinesis. First is karyokinesis. The word kinesis means, yes, the word kinesis means division. Karyo means nucleus. Karyo means what? Nucleus. So karyokinesis means the phase in which nucleus will divide. Nucleus will divide. That is called karyokinesis. Whereas the another phase is called, another phase, yes, what is that? Cytokinesis. Cytokinesis. We can divide the word into two, cyto and kinesis. Kinesis means once again division. And cyto means cell or cytoplasm. We can say anyway. Okay, cell or cytoplasm. So the cytoplasm division is called cytokinesis, and the nuclear division is called karyokinesis. Now, once again in karyokinesis, we have how many phases? Four phases. Okay, should I write here karyokinesis? So here I can write the short form. Okay, 1, 2, 3, 4. 4 phases. What are the 4 phases? PMAT. How to remember that? It's a key. PMAT. PMAT is the key to remember the steps in karyokinesis. Okay. Karyokinesis likra oru stage order ah நம்ம remember பண்ணிக்கிறதுக்கான key வந்து என்னது? PMAT. P for prophase. P for prophase is the first phase in the karyokinesis. Second one is metaphase. Metaphase. Meta means, the word meta means middle. Meta means middle. Anaphase. Ana means away. Ana means away. Anaphase. And the last one, telophase. Telo means end. Okay, telophase. So these are the 4 subphases present in karyokinesis. And what about cytokinesis? Cytokinesis is very simple. Cytokinesis அப்படினா cell divide ஆகிறது. இப்போ செல்லுல ஏதாவது ஒரு flavors இருக்கானா? Yes, there are 2 cells. One is plant cell, another one is animal cell. In case of animal cell, cell division, the cytoplasm division will be furrow method. It is called furrow method. What is that furrow method? Suppose a cell is like this. The cell will form a furrow, cleavage. Cell will form a cleavage and then cell will divide like this. This is called furrow method, and this is observed in animal cell. This is observed in animal cell. What about plant cell? So I'll take green color for plant cell. In case of plant cell, already the cell structure is like this, plate formation. Okay, so when the cell want to divide, there will be a plate formed in between the cell. That is called cell plate division. That is called cell plate division. So these are the 2 types of cytokinesis. In animal cell, there is no plate formation, whereas in plant cell, there is a plate formation. Next, we will see about interphase. Interphase is divided into 1, 2, 3. 3 phases, 3 subphases. What are the 3 subphases? Yes, G is called gap. So gap 1 and gap 2. These are 3 phases, gap 1 and gap 2. We can call it as G1 and G2. Whereas S is called synthesis phase, and very, very important. Most of the questions will come from S phase. Okay, S phase is also called as synthetic phase, synthesis phase, that is called S phase. Now, within this 3 phases, is there any subphase? Yes, we have one subphase within the G1 phase. இப்போ இந்த 3 phases குள்ள ஏதாவது ஒரு subphase இருக்கானா? Yes, answer is yes, that is in G1. Inside G1, we have one phase called G0 phase. We have what phase? G0. And if you read NCERT, it will be clearly given that G0 is quiescent stage. G0 is quiescent stage. Quiescent stage na enna appadina, if any cell is at rest, any cell don't want to divide, then the cell will be at G0 stage. Okay, G0 இருக்கிற cells எதுவுமே divide ஆகாது. The cells are doing its function. Already allotted functions அது செஞ்சிட்டு இருந்ததுன்னா அது என்ன phaseல இருக்கும்? G0. இப்போ divide ஆயிட்டு இருக்க cell எந்த stageல வேணாலும் இருக்கலாம். When a cell is dividing, no, it can be in any stage. This generally the interface is called as, generally நம்ம interface என்ன சொல்றோம் அப்படின்னா it is called preparatory phase. It is called preparatory phase. And M phase is generally called as, very good, dividing phase. This is called dividing phase. Okay, now we will represent it diagrammatically. How we can represent diagrammatically? We'll just draw one circle, and in the circle we'll represent, yes, here to here, it is called a M phase. This is called what? M phase. If you consider this as a cell cycle, the small portion is called M phase. Within this M phase, we can mark this is prophase, this is prophase, and metaphase. Next, anaphase. Next, telophase. Now this four phases together. Now this four phases together is called, very good, karyokinesis. Now this four phases together called what? Karyokinesis. So we'll use green color to represent this. How I can write this? KK, or karyokinesis. After completion of karyokinesis, that means now we are learning the order. Only after karyokinesis, which process will start? Cytokinesis. Now the rest of the portion we can mark what? Cytokinesis. Cytokinesis. Whether is there any type in cytokinesis? Yes, there are two types of cytoplasmic division. One is furrow division, another one is plate formation. Plate formation takes place only in plant cell, whereas furrow division is common in animal cell or cell without cell wall. Cell without cell wall. Okay. Now the rest of the phase is called, rest of the phase, what color will choose? Orange. Okay, the rest of the phase from here to here, it is called the orange. The orange color is called, yes, interphase. The orange color is called interphase. The interphase is divided into three. Yes, what are the three phases? Okay, you have any complaint that ma'am you are not using this color? Any complaint? In the color na use பண்ணவே இல்ல, இந்த color use பண்ணலாம்? Purple. Okay, we'll take this. Now this phase is called G1. This is called, yes, this is called G2. So in between somewhere we can show that quiescent stage. What is that quiescent stage? Yes, G0. So this is called a G0. G0 is present in G1. G0 is present in G1. So this is the introduction about cell cycle. Now we are going to discuss, first we are going to start with interphase. We are going to start with interphase. So we are going to learn what happens in interphase. So for that first we are going to learn from G1 phase. From G1 phase. So what happens in G1 phase? Because G1 phase la enna nadakணும் அப்படிங்கிறது எதை பொறுத்திருக்குன்னா, what is the next phase? Next phase is very, very important. What is next phase? S phase. So what happens in the S phase அப்படிங்கிறத படிச்சாலே G1 phase வந்து will be easy. S phase ல என்ன நடக்கும் அப்படின்னா, S phase ல very important thing, DNA replication. DNA replication, doubling of DNA. replication na என்னன்னு புரியலனா, replication na என்ன, DNA content double ஆகிறதுக்கு பேர் தான் என்னது, replication. Okay, so now in order to double the DNA, some proteins will be synthesized in G1 phase. So what will happen in G1 phase? Proteins will be synthesized. ஏன் protein synthesize ஆகணும்? Because அடுத்த ஸ்டேஜ் வந்து ரொம்ப இம்பார்ட்டண்ட்டான ஸ்டேஜ். ஸோ அடுத்த ஸ்டேஜில் டிஎன்ஏ டபுள் ஆகணும் அப்படின்றதுக்காக இந்த ஸ்டேஜில் ப்ரோட்டீன் சிந்தசைஸ் ஆகுது. அண்ட் ப்ரோட்டீனை ப்ரொடியூஸ் பண்ணுறதுக்கு we need enormous amount of RNA. Why RNA? Because from the RNA only we will get proteins. From the RNA only we will get proteins. Nityash, listen here. From the RNA only we will get proteins. Okay, so RNA also will be synthesized. RNA will be synthesized. Next, what will be synthesized? Some amount of energy, ATP will be synthesized. And because of synthesizing, the cell keeps on producing, cell will grow. Cell growth will occur, cell grows. Okay, so these are all the phases which these are all the process which occurs in G1 phase. Now if you check about this S phase, second one is centriole. Centriole doubles. Replication doubling, meaning is same. Okay, and centriole will divide. And here one important point to note, that can be a question. What can be a question? Centriole is present only in animal cell. Centriole is absent in plant cell. There is no centriole. Centriole is absent. Centriole is absent in what cell? So I take green. Absent in plant cell. Centriole is absent in plant cell. Centriole is present in animal cell. Is that clear? So this point, in this S phase, in the point எடுத்துட்டு வந்து plant cell க்கு எழுத கூடாது. ஏனா plant cell என்ன கிடையாது? Centriole கிடையாது. If it is animal cell அப்படினா தான் centriole duplication will take place. After this, histone proteins. Will be synthesized. Histones அப்படிங்கிறது, what is histone? Histones are the proteins present in chromatin. What is histone? Histones are the proteins present in chromatin. Next question, what is chromatin? So shall we take this color for DNA? Yes, DNA. So if this is DNA contains some beaded structure. We'll take yellow for, so like this, the beaded structure. So DNA will look like this. This structure together is called chromatin. In the structure பேர் தான் என்ன? Chromatin. புரிஞ்சுதா? What is chromatin? இப்போ இதுல இந்த orange colorல போட்டிருக்கிறது DNA. This is what? DNA. And this yellow one is nucleosome. This nucleosome ஒரு formula மாதிரி, max formula மாதிரி நம்ம convert பண்ணோம்னா, nucleosome equal to DNA plus histone. புரிஞ்சுதா? Did you understand? What is chromatin? Chromatin means DNA plus nucleosome. What is chromatin? DNA plus nucleosome. What is nucleosome? DNA plus histone. Easy? Okay. Whether histone is present in plant cell? Yes. Histone is common for all the cells. Okay. Histone is present in both plant cell and animal cell. So point to be noted here is only centriole fellow. This fellow will not be there in plant cell. It is present only in animal cell. Okay. So now in NCERT, if you check, they would have given C value. Given what? C value. So what is C value? C value is amount of DNA. What is C value? Amount of DNA. Okay, amount of DNA. For that we have to take chromosome. So we are taking, this is centromere. This is called centromere. Over the centromere we have chromatid. This is called what? Chromatid. Chromatin is different. Chromatid is different. This is chromatid. This is centromere. Inside chromatid only we have chromatin. Inside that only we have chromatin. So we'll take this color for chromatin. So this is DNA. Together we can call this simply as what? DNA. Okay, DNA. So now if we count this, for example, I will write here. Okay. Now the question is, a human cell is divided into two types. Human cell itself divided into two types. One is somatic cell. Somatic cell means body cell. Somatic cell means body cell. Example, liver, eyes, ear, kidney, nose, skin, anything. Okay, any body cell is called somatic cell. What about the other type of cell? Sex cell. Other cell is called sex cells. Okay, sex cell means it is present in gametes. Gametes are called sex cells. Gametes are called sex cells. Okay, now if you count the number of chromosomes, if you count number of chromosomes in your body cell, you are counting number of chromosomes in body cells. If you count the number of chromosomes in any body cell, you will find 46 chromosomes. You will find how many chromosomes? 46 chromosomes in your body. I am talking about your body. In your body, if you count any body cell, what is body cell? Other than sex cell. Other than sex cell. What is sex cell? If you are a girl, then egg is the sex cell. If you are a boy, then sperms are the egg cell. Sex cell. Sperms are the sex cell. Okay. Now, if you count the cell which is present other than any sex cell, you will have 46 chromosomes. If it is a gamete, yes, if it is a gamete, then it will have only 23 chromosome. It will have only 23 chromosome. If it is an egg, what color will take for egg? Blue or white? Okay. If it is egg, okay. If it is egg, how many chromosomes we will have? Same, 23 chromosomes. 23 chromosomes. Okay. This is egg or ova. Egg or ova. This is called sperm. This is called sperm. Okay. Now what we are going to learn, how these 46 chromosomes are maintained in all the cells. How 46 chromosomes are maintained in all the cells forever. Until we die, the 46 chromosomes will be maintained eventually in all the body cells. Okay. Next we will see in gametes how 23 chromosomes are maintained. To learn about this, we will read meiosis. To learn about this, we will read meiosis. To learn about this, we will read mitosis. Okay. Now from this image, you say me, which produces so many flavors? In the rendu division la, endha division la pudu products varum? Meiosis. Why meiosis? Because meiosis means the meiosis இன்னும் சிம்பிளா எப்படி புரியுற மாதிரி சொல்லலாம்னா how the parents are producing their children, offsprings. Okay. இப்போ parents மாதிரியே இருக்காங்களா அவங்களோட progenies எல்லாமே? நீங்களா அப்படியே பார்க்கிறதுக்கு ஜெராக்ஸ் மாதிரி இருக்கீங்களா? அப்படியே அம்மா அப்பாவோட even outside people அவங்க சொல்வாங்க அப்பா மாதிரி இருக்கா அம்மா மாதிரி இருக்கா. But we never feel like that. Yes or no? We feel that we are different. அவங்க எப்படி இருக்காங்க நான் எவ்வளவு அழகா இருக்கேன் என்ன போய் அவங்கள மாதிரி இருக்கேன்றாங்களே. Like that we used to think, right? So we never accept that நான் வந்து அப்படியே அவங்கள மாதிரி தான் இருக்கேன், அவங்க சித்தி மாதிரி தான் இருக்கேன், சித்தப்பா மாதிரி தான் இருக்கேன். We never accept because it's real actually. Whatever you are not accepting, because you are seeing yourself and you are feeling yourself, you are not similar to anyone. Okay. The reason for that is meiosis. Reason for that is meiosis, because the chromosomes will be shuffled and the new product will be created as a result of meiosis. But your skin, if you see, from LKG you are looking your hand, or from the birth you are looking your hand. Anytime did you observe any changes in your palm? Whether you are observing that, oh my hand today I found that இது வந்து நல்லா வளர்ந்துருச்சே பரவாயில்லையே அப்படின்னு anytime you felt your growth? No, it is like simultaneous process and you cannot differentiate any cells. New cells edu old cells edu differentiate பண்ண முடியுதா இங்க? இது வந்து new cell, இது வந்து old cell. No, you cannot differentiate because all the products are identical. All are identical to each other. Here the products are not identical. So we are going to learn how meiosis takes place and how mitosis takes place. But at the meiosis or mitosis or DNA duplication will take place only once. DNA double only once. Why I am saying that? Because in case of mitosis, we'll get only 2 daughter cells. We'll get only 2 daughter cells. 2 daughter cells. But in case of meiosis, we will get 4 daughter cells. We will get 4 daughter cells. So 4 daughter cells இருக்கிறதுனால S phase ல எல்லாமே டபுள் நடக்குமா அப்படின்ற ஒரு டவுட் நமக்கு நார்மலாவே வரும். Because meiosis 2 தடவை டிவைட் ஆகுது, மைட்டோசிஸ் ஒரே ஒரு தடவை தான் டிவைட் ஆகுது. ஸோ ஆப்வியஸாக நம்மளோட மைண்ட் என்ன நினைக்கும், டிஎன்ஏ அங்கே டபுள் டைம் எல்லாமே டபுள். நோ, all will take place only once. Okay, DNA duplication once, centriole duplication once, histone proteins once. Okay, now we are going to see this DNA duplication. Now this is common for both mitosis and meiosis. Which is common? DNA duplication. Okay, now we'll see how the DNA duplicates. So this structure is called what? This is called centromere, and this one is chromatid. This is chromatid. How you will count number of chromosomes? Number of centromere. Number of centromere. Okay, now this is the chromosome. We have taken for DNA orange, right? Okay, this chromosome is going to change itself in S phase. So what are the changes the chromosome going to acquire? It is going to get 2 copies of DNA. The copies of DNA, okay, I can take another color for the new DNA. So this is old fellow and this is the green one is new. Okay, so how this will happen? So first the DNA is doubled. DNA is doubled. The orange one is old one and the green is newly produced. Okay, இது எப்படி மேஜிக்கா டக்குன்னு ப்ரொடியூஸ் ஆச்சுன்னா? நோ, G1 ஃபேஸில் இதுக்கான ப்ரிப்பரேஷன் எல்லாமே நடந்தது. ஸோ G1ல நடந்த ப்ரிப்பரேஷனை வச்சு தான் இங்கே என்ன ஆகுது? DNA வந்து டபுள் ஆகுது. ஸோ DNA டபுள் ஆகணும்னா என்னென்னலாம் வேணும்னா, we need enzymes, we need nucleotides, proteins, then ATP, very good. Energy is needed to double the DNA. Okay, next what will happen? So here we can see, here if the content of DNA, see listen, here if the content of DNA is 2C, then here we can write the amount of DNA is 4C. That's it. Here the amount is 4C. So 2C is doubled to 4C. But if you write the chromosome number as 2N, if the chromosome number is 2N, then here also chromosome is 2N. Chromosome is 2N. So what you understood? Only the amount of DNA doubled, the chromosome number remains same. Clear? Yes. Next what will happen? That chromatid. See the chromatid. Yes, the chromatid will start splitting like this. This is old and the green one is new. So now slowly what will happen? Okay. So now whether we are going to change the number of chromosome, can you say number of chromosome here? 2N. What is the amount of DNA? 4C. What about here? 2N, 4C. Why we are not changing the number of chromosome? If you see, the number of centromere is 1, 1, 1, 1. So number of centromere is not getting changed. So we are not going to change the number of chromosome. So centromere oda number change agalana, chromosome oda number change ஆகாது. And you can see the chromatid initial S phase க்கு முன்னாடி ஒரே ஒரு chromatid தான் இருந்தது, but here the chromatid is doubled. Okay, so this is what happens in S phase. Okay, here we will stop and we'll continue in the next class. Okay, thank you.